thank you from the world for everything that the Chan Zuckerberg Initiative and the BioHub have done during this pandemic. From working with the health centers around the globe, the rapid sequencing efforts, the in-country pathogen detecting, the CLIA certified lab, all of these work combined together. This is a new era of molecular epidemiology, and it's what we're going to need for the next pandemic and more going forward. And it's just exactly that kind of large scale, immediate, collaborative data and information sharing initiative that is what the world needs as our health concerns are increasingly global and increasingly immediate. So I am both a pandemic virologist, structural virologist, and also the president of this institute. That's my research lab through the window. And I'm here today on behalf of a very large consortium called CoVIC, the Coronavirus Immunotherapeutic Consortium. This consortium is funded with equal parts public and private funding from NIAID, the Gates Foundation, and the GHR Foundation. And what this large collaboration set out to do at the onset of the pandemic was to look at antibodies against it that could be used for prevention, for treatment, to inform vaccine design, and understand what were the best antibodies against this virus, how we would define best, what criteria do we use in vitro and in vivo to decide what's better than another, what combination might be best with the understanding we probably need a cocktail to mitigate escape, and how do we make an antibody treatment that is affordable to everybody in the world and not out of reach. Next slide, please. And so the idea for that kind of large-scale multidisciplinary collaboration to compare things side by side came from, next slide please, work we did in the Ebola virus pandemic beginning in 2013. Next slide please. Typically, what had always happened in our emerging infectious disease field for understanding antibodies against this virus is that one or a couple of labs working together would make a panel of antibodies. And they would evaluate them on their ability to inactivate infection in cell culture, a term we call neutralization. Those that neutralize typically go into a mouse model. Those that protected mice go into a larger model. Ultimately, some go into non-human primates. Next slide, please. And the idea is that this pipeline that we had set up, that we did because we had to do, right? You can't put everything into primates. You have to choose them somehow. You can't even put everything you need into the mouse model. You must winnow them down some way. And hits came out the bottom. But every one of the steps in this pipeline can be viewed as a funnel where some things fall through and other things do not. And the literature only examines the things that fall through, and we have not fully examined the things that didn't. And so there's a formal possibility about something that could be protective in a living thing but didn't register as interesting in vitro, or something that could have protected in non-human primates but didn't have the activity you're looking for in the cell culture dish or in the mouse. And we don't know because they've wound up in the cutting room floor. And we would postulate that what is the least common denominator between each one of these funnels in our discovery pipeline is mechanical neutralization. The ability of an antibody to mechanically block the ability of virus to infect by either binding to the receptor binding site or blocking conformational change or blocking a required cleavage event. Those mechanical activities of Fab binding to spike are in common among tissue culture dish, rodent, primate, human. But other more complex activities that an antibody brings through its FC, any of the 20 of different functions by which it recruits immune cell help, clears pathogen, and so on, these are more complex. These are transient, these occur in concert. They are different between humans and non-human primates, different again in rodents, and they may not have been assayed at all in the in vitro systems. Next slide. Next slide, please. And that kind of hit our field in the face in 2013 when this was the best results that the Ebola virus research field had. The most potent neutralizing antibody we had, crystal structure shown in left, orange Fab onto the gray Ebola glycoprotein trimer, neutralized beautifully in cell culture, but failed to protect the non-human primates. At the same time, USAMRIID had put together a cocktail of 3 antibodies, purple and blue, that together would protect non-human primates, but didn't neutralize. Next slide, please. And so we had a disconnect between in vitro success and in vivo success, and clearly we were missing something. What were we missing? Was this data telling us that we needed a cocktail, that a monotherapy wasn't going to work? Was this data telling us that we're missing an activity of the base of the IgG, the base of the antibody, the Fc that recruits immune functions? And that clue came from the cell type that you produced your antibody in determined its protection. And the cell type determines a glycan that's on the Fc, which determines how well it binds to and recruits different Fc receptors. The other thought was maybe neutralization wasn't the only criteria we should be examining, or we were evaluating it the wrong way. Next slide, please. And so the idea was that we together as a research field needed to rapidly figure out what it was that did lead to protection in living things, and we needed a statistically significant pool of samples in order to do so. We had looked at just a handful of antibodies because the field was small and many people had their samples kept to themselves. Maybe we had only found the exceptions and not the rule. Next slide, please. And so the question is, how are we going to do this? It was clear to all of us that we needed better tools. We needed to examine more samples in order to make sure that we were getting a good understanding of the things that would contribute to protection. And the challenge was every lab was working in a silo and keeping their samples and their information to themselves. And there was a pandemic which had begun, which was threatening to escalate. And so rapidly we needed to do something. We needed to find a way to build a research framework that would enable the collaboration that we all know we need, would permit the sample and data sharing, but it had to function within a research environment that is fundamentally built on competition. Competition drives biomedical research. It inspires us to get our results faster. Competition drives innovation. Competition invites you to do a better job because your results must stand scrutiny. And yet, we also know that we must have collaboration. We must share tools, we must share resources, we must share information. We need many minds in a problem, especially when something is too big for any one group to have the tools to address alone. And that's a bit of a challenge, because even if investigators wanted to share their information or share their antibodies, maybe the university wouldn't let them. There was intellectual property involved. Or maybe they were torn between publish or perish versus wanting to build a better data set together. So how could we actually do this? We thought about this, we went through a lot of iterations. We came up with a study by which all of the antibody samples were blinded. They're all given a code name. That made it very fair. Everybody worked on the same set of samples contributed from researchers around the world. Everybody sends their samples in. They're all given a code name to make it very fair. Identical box sets of the world's supply of antibodies are sent out to different participating labs to do whatever assay they would evaluate antibody activities on. They did it on the same set. We combined that data back together. We had agreements that would protect everybody's intellectual property. If we together found out that some antibody was fantastic, that was just good for the owner, they owned everything at the end. And then we also wanted to make sure that we addressed the challenge with building a large group. Speed. The time it takes to organize a group can compromise the speed with which you can come up with results and publish a paper. We know we needed two things. We needed speed in the pandemic, and we needed a broader, deeper study. So we did a two-track study with a good idea and a limited set of people to rush forward to put something in place, and a broader, deeper study to move along and come up with the broader, deeper, more lasting information. Next slide. And so what that effort did is it launched a global collaboration with ultimately 44 mostly formerly competing labs across 5 continents, academic, industry, and government, working together into a single comprehensive effort to figure out what antibodies were best and how we might find them better in the future. Next slide, please. We studied 200 monoclonals across 3 years, about 100 people working together. And the antibodies that we looked at are in the vertical column and everything we measured about them in the horizontal column. Next slide, please. And we had so much data that we could start to make network correlations to see which features of antibodies tended to track with other features, which features were linked to protection. Next slide, please. And then we could, at the end of the day, build a regression to understand what are the features that you can measure high throughput in vitro that together predict protection. So a big bar is a strong predictor, blue is a good feature, red is a bad feature. And if you put these things together and maybe you only need to do the most important ones, we could predict protection in the mouse model with a 96% accuracy of these things measured high throughput in vitro. And what we find is that neutralization is indeed among the most important predictors, but it matters how you do it. The right shape and structure of the virus and presence and absence of a secreted form of the glycoprotein are key. Another indicator that we hadn't previously considered as much was the fraction of virus left unneutralized. But then there were a lot of other things that we only found by having this broader, deeper data set with lots of odd one-off antibodies that people would have otherwise sort of thrown away. And that's the importance of recruiting FC functions and building the immune system to clear pathogen. Next slide. And so at the end of the study, and that was 5 years, the group together put out 78 papers and conferred 10 PhDs in the results of that consortium, and revised the research pipeline, and then members of the consortium put 6 new therapies in place where there hadn't been any before. Next step. So we were asked in 2020 to pivot that kind of framework to SARS-CoV-2, to analyze things side by side and figure out what's best and what the best cocktail going forward might be. We now have over 50 collaborating labs and companies and over 370 different competing antibody therapeutics. And each one of these was discovered differently. Everybody discovery engine had a different strategy. Some looked at SARS-CoV-2 survivors. Some antibodies came from SARS-CoV-1 survivors. Some came from llamas. Some came from alternate biologics. Some were engineered in silico. So everybody had a different discovery strategy and a different evaluation strategy, which helped us get a broader sample pool to analyze. Next slide, please. Our contributors are about 2/3 industry, about 1/4 academia or nonprofit, and a few government, off 4 continents. Next slide, please. And among the industry discovery engines that contributed therapeutic candidate products for us to evaluate, many are from small and startup companies. About 8% of our panel are from the major multinational corporations and the household names. And so you can see the advantage to the startup companies and the small companies is that in this study, all of their molecules get an equal shot or evaluated on an equal playing field from the ones developed from a major corporation. And then a small company has access to lots of different kind of data they wouldn't have access to themselves, and they get all that back. Next slide, please. This is what it looks like from our standpoint in the laboratory. Everything's blinded, all candidates are on an equal playing field. And so the antibody therapeutics you've read about in the news are somewhere in there. Next slide, please. I'm blinded. So the samples come in, they're blinded, we aliquot and distribute them to different partners. We do functional analysis in parallel, looking at how they bind spike, how they compete with each other, how they neutralize in different systems, whether they recruit FC functions or structures or propensity for escape. All of the data gets put into an immediate publicly available database at covic.lgi.org, and you can just download all that data into your own Excel spreadsheet. And we're also analyzing the things being evaluated with the clinical trials side by side for parallel independent data. Next slide, please. Okay, so the first thing we did was take all of these antibodies and bin them by their degree of competition. So we have a cluster that hit a footprint that we're coloring orange and a cluster that hit a footprint we're coloring yellow. They're against different sides of spike. Here are 7 major groupings against the receptor binding domain. Next slide, please. And so other studies analyzing and sorting antibodies against spike have separated them by germline or by visible structure. Here we're separating them by degree of competition. And as you might expect, there's a lot of alignment and similarities between the visible structure and the degree of competition. So we can divide them into major groups and subgroups based on where they bind on spike. Next slide, please. And we can have initial images. This is negative stain microscopy looking at those in category 1. How do they approach and hit spike? What is their footprint? They have remarkably similar approaches, even if they come from different germlines, different species and a footprint. So groups 1, 2, and 3 hit the receptor binding motif, exactly where spike binds receptor. 4 and 5 hit the outer face of the spike, 6 and 7 the inner face of the spike. So spike is like this. The receptor binding domains begin down. In order to bind a receptor, they lift up. That is the receptor binding motif. Some antibodies neutralize potently by binding exactly there. Some bind the outer face of spike, so they can bind it when it's down or when it's up. Others bind the inner face of spike. They can only bind it when it's up. Next slide, please. And then we can look at the footprint of each of these major categories and subcategories and look at how well that footprint overlaps with the binding side of receptor. Binding side of receptors in a dotted line, the footprint of each group of antibodies in color. So group 1 nearly precisely overlines with that dotted line receptor binding site. Group 2, potent neutralizers slipped a bit this way. Group 3 slipped a bit that way in yellow. Next slide, please. And then there's some that are slipped entirely out of the receptor binding site. Now the footprint that they bind tells us their susceptibility to the variants to a large degree. And that was another reason why we wanted to do this kind of analysis. Next slide, please. In addition to looking at the FAB, we also looked at how the whole IgG would interact with spike because we were interested in steric effects and stoichiometry. So for example, group 1 tend to fully occupy spike with 3 different IgGs bound at the same time, 1 Fab into each receptor binding domain. Group 2, some of the potent neutralizers, and group 2 contains a lot of the therapeutic candidates you've read about in news, tend to bind bivalently, both FABs from one IgG anchored as goalposts with the FAB in between. They bind 2 of the receptor binding domains, the third is left unoccupied. Group 5 in turquoise, these are interesting. They don't bind the tip of the receptor binding domain. They bind slip down the side. And they're slipped down the side enough of the spike that a lot of what these guys do is cross-link one spike to another. Now I'm showing you a couple of cartoons in the bottom. One is where 2 spikes oppose each other and the IgG is cross-linking. This would not happen in biology. This happens with soluble ectodomain and an EM grid. I suppose it could happen if 2 viruses oppose each other. What is realistic in biology are the 2 spikes that are cross-linked by this. That spike on the surface of the virus can have a 40-degree rotation either way. We see a lot of the group 5 antibodies cross-linking 2 spikes together, 1 IgG hitting 2 spikes, and those that neutralize most potently in group 5 are the ones that cross-link. They also have some of the best in vivo protection at consortium with complete protection in the mouse model at very, very low doses. So we can categorize their footprint and how IgG interacts with spike. Next slide, please. And that tells us a lot. Their footprint tells us how or whether they are susceptible to the different variants of concern. So when the variants started emerging, we rapidly had to expand and pivot the study to find candidates that would be durable through time. So here's your view of the coronavirus spike, viral membrane at the bottom, receptor binding domains at the top. In this guy shown here, 1 receptor binding domain is lifted up. Now I'm going to rotate that structure. Next slide, please. So you're standing on the target cell looking down into the receptor binding domain. Next slide, please. Here's just the 1 receptor binding domain with the location of each of the point mutations that have been linked to different variants of concern or have arisen by themselves with high frequency in the population. Next slide, please. If you draw an axis down the center to the inner face of the spike and the outer face of the spike, I can show you where the different footprints are and the different antibodies bind. Next slide, please. And we can come up with a competition grid across the study. So dark blue means that each one of these, this is the first couple hundred. Dark blue, these antibodies compete, light blue, they do not. So we can use this as a strategy for cocktail selection. Let me show you a subset of 50 of these that we evaluated for binding and neutralization to alpha through epsilon and multiple single point mutations in viruses that emerge from MINC. Next slide, please. So here's 50 example antibodies chosen to represent the breadth of sequence and structure space across the RBD binding motifs and the different groups and subgroups. You can see, for example, that those in group 2A tend to be knocked out by mutations at position 417. Those in 2B tend to be knocked out by E484 mutations. But if you look over on the right, the turquoise, blue, and purple, those footprints tend to be variant resistant. They are slipped down the side of the spike, away from the receptor binding domain, and they tend to continue to neutralize all the variants that have emerged thus far. Next slide, please. So for example, here are some of the more potent neutralizers that hit the receptor binding motif in groups 1, 2, and 3. You can see in the black boxes that many of those group 2 antibodies lose efficacy against the beta and the gamma viruses. Delta is different. And so whether the antibody is going to work or not depends a lot on what the mutations are. But for something that's going to be durable and last a long time, we want to find candidates that potently neutralize. So what I'm showing you here are lots. Each vertical line is a different antibody in the study, sample 69, sample 259, sample 186. The colored spot is potency of neutralization, how it's been affected by mutations in the variants. If it's turquoise, it neutralizes that variant better. If it's brown, it doesn't neutralize it at all. Gray, nothing happens in the middle, gradation of color. So you're probably interested in the molecule all the way over on the left, under number 1, CoVIC 69. That molecule are 2 copies of the ACE2 domain linked to a human FC. So it's an artificial construct, but because it's made from ACE2, it always binds, neutralizes those mutations that try to achieve better ACE2 binding better. It's not as potent as the natural human IgGs, but it's a good proof of concept. Next slide, please. What's interesting to us, and potentially a more durable therapeutic going forward, are those antibodies in groups 5, 6, and 7, and this other one in group 4, that tend to be resistant to whatever variant might want to emerge or the mutation against it toss out. These are, I think, good candidates to make cocktails that'll last a long time. That one, 268 in group 4, that shows you that there are individual examples in these groups. Although most of the groups might be knocked out by a variant, that guy's still a winner, and he potently neutralizes 7 milligrams per mil. Next slide, please. So that is what we have done in the first year, was to build the study, gather these, understand how all the different antibodies from the different discovery engines across the world compete, how we can make cocktails which are durable and resistant. In the next year, we're going to advance some of these cocktails and try to evaluate the predictive capacity of our different in vitro assays for predicting protection in the animal model. That database, all of the data is freely downloadable and accessible, and it's been built with FAIR principles, findable, accessible, interoperable, reusable principles. So the idea that this is probably one of the more lasting aspects of this study, the ability to have all of this different kind of information on the performance of different antibodies. That information on what antibodies are ideal and how we can try to shape spike to build better antibodies has allowed us to also engineer a 3rd generation vaccine candidate. Next slide, please. And we're showing you the cryo-EM of that structure here. We call that spike V-flip, V for Roman numeral 5. It has 5 stabilizing proteins. FL for flexible linker. We've added a flexible linker to tether the S1 to the S2. And IP, we've added an interprotomer disulfide bond in the core that holds it in its trimeric prefusion configuration. Its yield is very high. We routinely get well over 150 mg per mil expression. It is very stable. We can leave the recombinant protein in the lab drawer for a month at room temperature, and it's still trimeric in the right fusion. We have worked with Ju Snyder in the Netherlands to evaluate the carbohydrates. And because we think it has a better structured core, the carbohydrates that are attached to it better reflect those that are on the native virus. And we immunized mice to find out what the antibody response looks like. And we looked at level of binding antibody between the 2P that was in the vaccines that we all got, Hexapro and V-flip, equivalent levels of binding. When you look at neutralization, we first looked at 1 month, we get a higher titer of neutralizing antibody from this better stabilized spike. And then we sat on our hands and we bit our nails for 6 months to see what the response would be 6 months. That's what we want. We want a vaccine that's going to last a long time. And we find that after 6 months, the horses start to separate in the race, and that we do get a higher neutralizing titer from V-flip. And so we think we can probably expect that we will have recurring coronaviruses for quite some time in years to come. If we want to develop improved vaccines for seasonal boosters or new things, this is a good framework to do it with. Next slide, please. And a consortium of this size would never have gotten off the ground without our funders and our inspiration. The COVID-19 Therapeutics Accelerator, a supplement to the Ebola Consortium grant from NIH, the GHR Foundation, private support from the Overton family. And I especially want to thank all of the different people involved. This is a lot of heavy lifting to build something of this scale and this size. Most of my lab went over to trying to move this project forward, and I'd also like to thank all of our partners that either contributed their published and unpublished therapeutic samples or took on the burden of this work to build this database together to find out what we can learn going forward. And I'd like to thank you for your time and your attention.
Do we know if different COVID vaccines are likely to induce antibodies with different neutralization mechanisms?
They do. You would get a variety of antibodies that did different things. They could bind to the receptor binding domain and either directly block ACE2 or by occupying this footprint or sterically. There are antibodies that prevent cleavage. There are antibodies that likely prevent conformational change. There are lots of antibodies we may not have entirely figured out what they do next. And of course, there are antibodies that also recruit FC activities in building new complexes. So I think what she's actually asking is, does one vaccine format elicit a different kind of antibody from another?
Yes.
I don't know the detail to which that has been analyzed, but I do think that if you change the structure and form of the immunogen, you will change the surfaces elicited. So Studies in HIV and loss of virus have shown us that getting the right pre-fusion structure on the right oligomer is essential for finding the kinds of antibodies that bridge one subunit to the other. Often these quaternary epitope antibodies are among the most potent neutralizers. We also know that it's important for antibodies to fish down and find the right protein surface between the glycans. And it's important for the glycans to accurately reflect what's on the virus, because often the antibody has to thread a needle down in there, or else the antibody partially recognizes the glycan. The glycan needs to be the right structure. So I would hypothesize that something with a better glycans and a better oligomeric structure would elicit more of those kinds of antibodies. Antibodies against the receptor binding domain, I think, are a lower hanging fruit, and I think lots of different kinds of vaccines will elicit those better because it's easier to get the receptor binding domain folded well and presented well.
That's great. Now, for a virus like SARS-CoV-2, which in the end, during its route of infection, is in different anatomical and tissue sites, so in the nasal epithelium and then eventually in the lungs. That doesn't matter what antibody is that a case where the effector function of an antibody may make a difference in terms of its effectiveness in eliciting appropriate immune responses in a particular tissue or anatomical site?
I think it will. I think it matters very much what the antibody does and how it's delivered. So an antibody to prevent infection may need to be a different kind of thing from an antibody that neutralizes an already established infection. And an antibody that could be delivered by an inhalational route might have an easier time accessing respiratory tissues than ones that are delivered IV. And so one of the things that we're wanting to do is to compare different routes of administration to see how this affects the type of protection and the different viral loads in the different tissues. We don't have enough data yet, but this is the question that needs to be asked.
That's great. We then have the question, have you evaluated antibodies raised by the current common vaccines? And what are your thoughts on the debate around natural immunity from a person being infected versus the antibodies that emerge from being vaccinated?
Other people have done that work. This consortium is mostly antibodies from survivors. I think there were some from vaccinés. I don't know all of the sources because I'm blinded. The debates on natural immunity versus antibodies that emerge from being vaccinated, of course, the big detriment to natural immunity is you had to go and get sick to get it. So that's not ideal.
Yes.
And there's a lot of evidence that antibodies from the vaccines can be stronger and better with the really strong responses from people that had an infection and then got a vaccine. There is, of course, a lot of variability within an individual's response. And so there are certainly some of one category that are higher than others. I think the antibodies against the vaccines look pretty good, but I haven't analyzed them in detail in the same way as I've been evaluating the therapeutic delivery candidates.